All right, good afternoon, everybody. Thank you for attending today. This is the beginning of a new type of seminar series that we're doing with the U.S. Society for Ecological Economics. We're calling it Putting the Ecology Back into Ecological Economics. Came out of discussion with John Gowdy and Corinne Lindberg, and Corinne really helped facilitate this, so I want to give her a special thank you. And so this is really the first of those seminars. We have a lot of different topics coming up. Methane seeps. We have coral reefs coming up, grasslands, forests. So you should hopefully all be getting those announcements soon. Today, our speaker is Olivia Pereira. Hopefully, I pronounced that right. She's a postdoctoral scholar at the Levin Lab in the Scripps Institution of Oceanography. And today's talk will be making sense of methane seeps, ecosystem dynamics, and resilience and linkages. And she will also be we're fortunate enough, we found one person, kind of a unicorn, who's doing the ecology piece and the economics piece. So she'll also be our speaker on the follow-up with the economics talk next week, understanding and valuing human connections to deep sea methane seeps off of Costa Rica. So we can't thank her enough for doing both of these seminars for us. And without further ado, thank you, Olivia.
Thank you so much, John. Thanks for this introduction and for having me here today. I'm super excited. I am sharing my screen and presenter mode. There we go. Okay. So today's talk, let me also just remove this thing. Hide um video panel maybe? Nope. Hide floating meeting controls. Okay, awesome. So today, like John mentioned, we thought that it would be interesting if I could give an overview of the science of methane seeps first, so that you all can understand why scientists care about the deep sea, so that next week I can present the economic valuation study that I did with methane seeps, and we can see why people care about the deep sea and methane seeps. So when I talk about the deep sea, a lot of people tend to think that this is what it looks like. It's just this muddy bottom with not a lot of things happening down there, just some animals here and there. And deep sea scientists know that there's actually quite a lot of things happening down there in this video only. But this is what most of the deep ocean looks like. This is what the abyssal plain looks like. And even though most of the deep ocean looks like that, there is still ecosystems in the deep sea where we find hard ground ecosystems. So there are mid-ocean ridges, seamounts, canyons, chemosynthetic ecosystems, continental margins where we find phosphorite and ferromanganese rocks. And even on the abyssal plains, we still have some hard grounds like the polymetallic nodules. You might have heard about them because they're getting a lot of attention because of deep sea mining lately. But from these photos, you can see that all of these hard substrates attract a huge biodiversity of animals. So in all of these photos, we can see lots of animals and even colorful animals. These animals are using these substrates as attachment, as refuge, as reproduction, or as feeding grounds. And my research has been focused on methane seeps for the past few years. I've been working with chemosynthetic ecosystems actually for quite a while now, since my first year in college. Um, but it was on organic falls, and now I switched to methane seeps. And I'm going to explain a little bit more what chemosynthetic ecosystems are later. But first, I will talk about methane seeps, which is what my research has been focusing in the past few years during both my master's and my PhD, and now my postdoc position at Scripps. So methane seeps are chemosynthetic ecosystems, which means that they're fueled by chemosynthesis. Chemosynthesis is the process of microbes taking up chemical compounds in the water to produce organic matter, fueling this entire ecosystem. So, in shallower waters ecosystems like coral reefs, kelp forests, or any other shallow water ecosystem, we have plankton using nutrients and the sunlight to produce organic matter in the photosynthesis. In the deep sea at chemosynthetic ecosystems, we have chemosynthesis because there's no sunlight. So microbes taking up chemical compounds to produce this organic matter. So it is a very localized production of organic matter to these animals. Methane seeps are found worldwide on continental margins, like you can see here in this map, from depths at 20 to 70 meters, as shallow as 20 meters, to all the way down to more than 7,000 meters. And they play a really important role in climate regulation and carbon sequestration. Methane is a very powerful greenhouse gas, even more powerful than CO2. So by taking up this methane that otherwise would be released into the atmosphere and the water column and the atmosphere, sorry, methane seeps can be either a source and a sink of methane to the atmosphere. And methane seeps are found associated with hard substrates in the form of carbonates. So these carbonates are a byproduct of the microbial activity of the chemosynthetic production. So this is what the microbial activity looks like, the equation producing a lot of bicarbonate. This bicarbonate reacts with calcium in the water and forming all of these extensive formations of carbonate rocks. And you can see in this photo that there is already a lot of animals using these rocks. We have giant mussels. So all the brown stuff here are giant mussels. There's a giant crab over here. And if you take a closer look, you're going to see that there's also some smaller animals. So limpets over here, some snails, a shrimp, there's some worms. So this is a little worm tube. And these are the animals that I work with. So these are what we call the invertebrate macrofaunal community that we find associated with carbonates and methane seeps. And methane seeps provide a lot of ecosystem services. Ecosystem services are a very human-centered vision of ecosystems because they are kind of like benefits that we get from the ecosystems. So I mentioned already carbon sequestration, but also oil and gas for the energy industry, nutrient cycling, endemic species. So endemic species are species that are only found at methane seeps. And here we're thinking about in economics terms, this would be the non-use value and the biodiversity value that we have from these ecosystems. Seeps also provide nursery and feeding habitats for economically important fisheries, and they also provide cultural services. So art, education, inspiration for music and movies even, and also the spiritual connection that some cultures have to the deep ocean. And because seas provide all of these services and they're found on continental margins where it's kind of easy access, there is a few disturbances and threats to these ecosystems. So I mentioned that they provide feeding and reproduction grounds for economically important fisheries. So bottom trawling and bottom fishing is a big thing for methane seeps. Oil and gas extraction, like I mentioned it before, for the energy industry. And I think that when we talk about the ocean or when we talk honestly about any ecosystem in the world, we have to talk about climate change. And climate change has already reached the deep ocean. The deep ocean is already warmer, almost 2 degrees Celsius warmer. And that is quite a lot if you think that the deep sea average temperature is 4 degrees. So ocean warming is a possible threat to methane seeps because these gas hydrates are actually very unstable. And just a little bit of a change in temperature and pressure, these might dissociate and we might actually see an expansion of methane seeps. We might see more methane being released out of the seafloor. The other really important thing about climate change in the deep ocean is ocean deoxygenation. So the ocean is losing oxygen for various reasons that you see some listed here. But ocean deoxygenation is especially important for methane seeps because we also find oxygen minimal zones on continental margins where we find methane seeps. So oxygen minimum zones, or OMZs, are areas, coastal areas, where we have a lot of production of photosynthetic production in shallow waters because of all the nutrients that come from deeper, colder waters. This excess of organic matter that is not used in the shallow waters sink to the bottom of the ocean and is reworked by microbial activity that to do that is taking up oxygen. So it depletes this layer, this ocean layer in oxygen, and we find these OMZs. And with ocean deoxygenation, we might actually see an expansion of these OMZs. And this is going to have an effect not only in the communities that are living already within the OMZ, but also for the communities that are living above and below the OMZ, because they might see their habitat getting compressed with the expansion of this OMZ. And by studying the animals that we find currently within OMZs, we can try to have a better understanding what might happen in the future to these other communities if these OMZs really do expand. So the goal of my research for the past few years during my PhD research and my master's also was to characterize and understand the dynamics and resilience of these macrofaunal communities that live associated with carbonate rocks across environmental gradients. So we can inform response of these ecosystems to changes associated with both climate change and deep sea resource extraction. So for example, fishing and oil and gas, and provide insights for future management and conservation efforts. However, these management and conservation efforts are going to have a higher potential of success with increased public support. So, for the past couple of years, I incorporated socioeconomic approaches to my research to understand people's care about and valuation of the deep sea. So, this is a little bit of a sneak peek of what I'm going to be talking about next week when I'm going to actually talk about these economic valuation study that I did. But for today, I'm going to focus, like I mentioned, in the ecology and our ecological science of the dynamics and resilience of these animals. So my study area is the Costa Rican Pacific margin, and it is an incredible place for us to study methane seeps across environmental gradients because there is a well-defined oxygen minimum zone. So here I have a vertical profile of depth on the y-axis and oxygen on the y-axis in blue. And you can see that there is this very low oxygen layer here that is the OMZ at intermediate depths. And we find methane seeps across both the oxygen and depth gradients. So we have Kippos Landslide, Mount 12, and Hako Scar. These were three of our methane seep sites. Costa Rica is mostly ocean, so 92% of the territory is ocean, and they're very committed to marine conservation. So it's been really an honor and a privilege and such an amazing opportunity to be working in Costa Rica and with Costa Ricans and learning from them and how they respect their marine environment and the marine ecosystems. So these were the three methane seeps that we visited in Costa Rica. Quepos Landslide at 400 meters within the OMZ, Mount 12 at 1,000 meters just below the OMZ, and Hako Scar at 2,000 meters, well below the OMZ, so highly oxygenated, but it has a little bit of a thermal anomaly. So there is one location at Hako Scar that has temperatures of 8 degrees instead of 2 degrees. So you can see from these videos that these methane seeps look very different. And we wonder what is causing all of these differences. Is it seepage activity? Is it oxygen? Or is it depth? So I want to just talk a little bit also about the seeps here and tell you what you're looking at. So Kippos landslide, so the one here on the top, you're seeing a lot of microbial mats. So the white stuff on the seafloor, there are microbial mats. And the dots are tuna crabs. So these are crabs that just show up one there and they are just taking over the entire place. Mount 12 on the bottom left is covered in very extensive formations of carbonates. And we have a lot of the giant mussels that you see, the brown stuff. And in the back, we have the Yeti crabs. And these are endemic species to Mount 12. Mount 12 is the only place in the world that we find that species of Yeti crab, as far as we know. And Hakkasgar doesn't have as many rocks, but it has these huge tube worm bushes. So this whole thing here, they're tube worms. They are little worms, and it's also covered in a lot of clams. So they're very different. To visit all of these sites, we went to Costa Rica for a few different years with the RV Atlantis, the research vessel Atlantis, and the submersible Alvin that you're seeing in the middle. And this is a photo from inside Alvin. I got to dive in Alva to Mount 12 and see the Yeti crabs out of the window. And we also went in 2019 with the research vessel Falcor and the ROV Sebastian that you're seeing in this photo. We use Alvin and the ROV to collect the rocks and all the other samples that we collected with the manipulator. So the arms over here, we put the rocks in these white boxes and we bring them up to the lab. And this is what the rocks look like up in the lab. And you can see already that there's lots of animals here that live associated with these rocks. So we have some yeti crabs, baby yeti crabs, little snails, the giant mussels that I mentioned. This is a brittle star. So there's quite a few animals that live there. I pick all these animals, I sort through them, I ID them to species level, and this is what the invertebrate macrofauna community associated with carbonate rocks look like. We have a lot of mollusks, and I'm going to be talking a lot about these guys here. So they are what we call snails and limpets. They are feeding on the bacterial community on the rocks, so the actual fresh chemosynthetic organic matter from the bacteria. We have annelids or worms, crustaceans, cnidarians, and echinoderms. So I separated this talk into three parts: the dynamics, the resilience, and the links of methane seeps. So first, we're going to understand how these macrofaunic vertebrate communities change across seepage activity, depth, and oxygen gradients, looking at the natural dynamics both spatially but also temporally. Then I'm going to talk about the resilience of these communities across these environmental gradients, where we conducted some experiments that allow us to see if these animals would survive changes in these environmental gradients. And finally, like I mentioned, I'm going to link deep methane seeps to other chemosynthetic ecosystems. Okay, so first, let's look at the natural dynamics of these communities across seepage activity, depth, and oxygen gradients. To do that, we collected rocks at the three methane seeps and across the seepage activity so that we can look at changes in the communities along a seepage gradient and also along a depth and oxygen gradient when we compare the different methane seeps. These rocks are what we call the in situ rocks, so the natural rocks. And first, we're going to talk about the seepage gradient. So we're just going to look at the rocks that we collected at Mount 12 along the seepage gradient in different years so we can look at both natural, spatial, and temporal variability. So we're going to start simple. And I just want to explain a little bit more about the seepage activity here. So we separate these seepage activity levels just by eye. So we look at the site, we look at the place, and we decide if it's an active site, a transition site, or a background site based on visible cues. So active sites are areas where it's the main area of the seep is where there is active methane seeping out of the floor. We have the chemosynthetic production. We have formation of carbonate rocks. We have lots of bacteria, and we have these animals that are endemic to methane seeps. Background sites are the surrounding deep sea. So it's just the area where there's no seepage activity, there's no chemosynthetic production, and there's no formation of rocks. And transition sites are the in between. So there are areas where there's some rocks here and there, but there's both endemic active seep fauna and background fauna, excuse me, coexisting in this place. So let's look at some results. First, I'm going to show you community composition on the left side and density on the right side along a seepage gradient. So the x-axis here represent the different seepage activity and the different years. And remember that now we're just looking at Mount 12. So we see first that in 2017, active sites were dominated by these snails and limpets that are feeding on chemosynthetic production. It also had the highest density values in 2017 at active sites. In 2018, the community was still had still more of these snails and the limpets compared to transition sites. However, it was much less than in 2017. And if you're looking at this plot, you might be seeing that this bar here looks a lot more like this bar here, and the same for the densities. So we started looking at this and we wonder what is happening here that in 2018, the active site looked a lot like a transition site from 2017. And we questioned if maybe there was a decline in seepage activity from 2017 to 2018 that could explain this difference. And there were a few things that showed us that that was possibly the case. We found a lower abundance of these, especially these limpets that are feeding on the chemosynthetic bacterial community. So we call them bacterial grazers in 2018. And we also found a higher reliance on photosynthetic production in 2018. We know from the literature that natural variations of water column can happen within as little as 1 year, and that earthquakes can modulate and change methane seepage activity. We looked back and we saw that there was actually an earthquake that happened right before occurs in 2017. So it could be that in 2017, the seep was very active. And then in 2018, it wasn't as active because of that earthquake. And we saw this change in the community. So what we're seeing here is that these invertebrate communities are very dynamic. They change along a seepage gradient, but they also respond to a natural decline in seepage activity within just 1 year. So that is a very quick response that we're seeing for these communities. So now that we understand how these communities are changing across seepage activity and with ears, I'm going to complicate things a little bit more and introduce other variables. So here again, I have the vertical profile of death on the y-axis with oxygen on the x-axis in blue and our different methane seeps. Kippel's landslide within the OMZ, Mount 12. This is wrong, I'm sorry. Mount 12 is actually outside of the OMZ. This OMZ should stop above 1,000 meters. And Hako scar at 2,000 meters well below the OMZ. And low oxygen is a very important variable in the deep sea because it is known to cause a lower diversity, density, and biomass of animals, and that it tends to homogenize these communities. However, most of these studies with low oxygen have been done with megafauna, so animals that are a little larger than the ones that I work with, and on seamounts, so different communities, different ecosystems than methane seeps. And there's still a lot that we don't know about how low oxygen is affecting chemosynthetic ecosystems, and especially the macrofaunal, the small little animals that I study. The other variable that we're going to look at is depth. And depth is important because there is a less export of photosynthetic production with increasing depth. So the deep sea, the abyssal plain, for example, depends entirely on food that is sinking from shallower waters, on the photosynthetic production from shallower waters that sinks down to the bottom. At chemosynthetic ecosystems, we also have that local production of organic matter in the form of chemosynthesis. So shallower seeps have this mixed influence of photosynthetic and chemosynthetic production. But with increasing depth, we expect to see that deeper seeps will show a higher reliance on that local chemosynthetic production because not as much photosynthetic production reaches deeper depths. So to look at how depth and oxygen influence these communities, like I mentioned before, we're going to compare the different methane seeps. We collected rocks along the seepage gradient at the 3 seep locations so that we can see how also depth and oxygen are influencing that seepage activity influence on these rocks. Looking first at the natural dynamics across depth and oxygen gradients. So here we're comparing the different seep locations, Kippos to Mount 12 and Hako scar. On the left, I have a community composition plot, and on the right, a density plot. And the x-axis here represent the different methane seeps, so Kippos, Mount 12, and Hako, and different seepage activity. And we see that Kippos had more of these hydroids, the little photo here, which are little cnidarians. Mount 12 is dominated by those nails and limpets that I already talked about. And Hako scar is dominated by these little crustaceans that are called tanaids that you see here in the photo. Looking at densities, Aquascar showed the highest densities. What was interesting about that is that that was mainly because of the high abundance of these little crustaceans. Tanaids are known to have an increasing abundance with increasing depth at just overall ecosystems. So it's interesting to see that this is also the case at Methane Seeps, so within chemosynthetic ecosystems. The other interesting thing that we saw when comparing the different methane seeps and looking at changes in depth and oxygen was that Mount 12 and Hakuskar, which are seeps that are outside of the oxygen minimum zone, are super similar. So this is a cluster analysis, and the closer together these little clusters are, the more similar these communities are. And it's interesting to see that they're very separated into the methane seeps. So Mount 12 is mainly here, Hakuskar here, and Kippo's landslide here. So Kippo's, it's actually the most different seep from all of those three seeps, and it's the one that is within the oxygen minimum zone. We also observed that in the food web. So I'm not going to go into too much detail about how we study food web and trophic structure, but that was another part of our analysis. But I think it's interesting to see that Mount 12 and Hakoskar had these highest overlap in food web structure, meaning that these animals are feeding on more similar things. In Kippo's landslide, the animals are feeding on something else. We also saw with this data set that animals at Mount 12 and Hako, especially at Hako, have a higher reliance on the chemosynthetic production, as we expected, because there are seeps that are found at deeper depths. While at Kippos, our shallowest seep, it actually relies on both photosynthetic and chemosynthetic production. Now, looking at all three variables, we're going to look at seepage activity, depth, and oxygen gradients. So looking at within each seep, if seepage activity is still having an influence at these seep locations. And we see that I mentioned already that Mount 12 changed with seepage activity, and we find less of these snails and limpets at transition sites. At Hakoskar, we also saw there was a changing community, and the tanaids are the little crustaceans are actually more abundant at transition sites. We also see some differences in densities with seepage activity. However, Kippos, things were a little different. And these differences that we see in these plots were actually not statistically significant. And because this seep is the one that is within the low oxygen conditions, we hypothesize that low oxygen overrides the effects of seepage activity. So the differences between active and transition are not as clear as in Mount 12 and Hakuskar. But when oxygen is not a limiting variable, the community composition, density, and food web structure change with seepage activity and depth. Okay, so I explained the dynamics of these ecosystems across all of these environmental gradients, and we know the natural dynamics of these animals. But what about the resilience? We know that oxygen might be playing an effect into the dynamics of these animals, but what will happen with these communities in the case of ocean deoxygenation and ocean warming? Are they going to survive? Are they actually going to start looking more like hippos, for example? So to look at resilience, we conducted a couple experiments. We used the in situ rocks that I talked about in the dynamics part of this talk as controls for these experiments. So we did a colonization experiment for different periods of time. The colonization experiment, we had rocks with us on the ship that were previously dried and didn't have any animals on them. We deployed them at active sites and at transition sites. And the goal of this experiment is to look at colonization patterns. So if we put a substrate down there, are these animals going to be able to colonize these rocks and take them up and reproduce and thrive on these rocks? We did this experiment at the three seep locations, so we could also see how death and oxygen are influencing these colonization patterns. The second experiment was a transplant experiment. The difference between the transplant experiment and the colonization experiment is that the transplant experiment, we basically just moved rocks around down there. So we went down with the sub, with an empty basket. We collected rocks with all the animals on them at active sites, and we moved them to transition and background sites. So this is what would be the idea of a seep dying. So if there's seepage cessation, what would happen to these animals? Would they survive at lesser seepage activity? We did the same thing from transition to background, from transition to active. So this is the opposite direction, and from background to active. So the idea of this transplant experiment is to look at the recovery and resilience rates of these animals. So like I said, if there's anything that happens with seepage activity, and we did see in the first part of the talk that seepage activity can change within just 1 year because of the natural dynamics of the ecosystem. So will these animals survive if there's less seepage activity or if there's seepage activation? And we did this experiment at the three seep locations, so we could again see how death and oxygen are influencing the recovery and resilience of these animals. So first, I'm going to show you the transplant and colonization results from Hakkasgar and Mount 12. So we're going to focus on seepage activity and death as our environmental variables for now. So with the transplant experiment, we saw that seep fauna and transition fauna, so that the animals that we find at the active site and the transition sites respond differently depending on depth. So at Hakkasgar, our deepest site, where we have a higher reliance on chemosynthetic production, it seems that the active fauna, so the seep fauna, is more dynamic and respond faster to changes in seepage activity. At Mount 12, where we are at an intermediate depth and we still have a little bit of an influence of the photosynthetic production, the transition fauna that is more used to those mixed conditions of photo and chemosynthetic production is more dynamic and responds faster. With the colonization experiment at both Mount 12 and Hakkasgar, we saw that these bacterial grazers, so the snails and the limpets that are feeding on the chemosynthetic production directly, so the microbes themselves, were the most successful colonizers. This is super interesting because Hakkasgar actually doesn't have a lot of these snails and limpets naturally, but they were very successful even at Hakkasgar in colonizing these rocks within 3 months, 17 months, 20 months, and 7 years. So not only in the short term, but also in the long term. And with these experiments, what we see is that the reliance on chemosynthetic production seemed to control the colonization and resilience of these animals at these two seeps. And I want to remind you that chemosynthetic production is actually associated with active seepage and it increases with death. So even this reliance on chemosynthetic production is still associated with our environmental variables, seepage activity and death. But what about Kippo's landslide? Things were a little different at Kippo's, and possibly because of low oxygen. So here we are seeing actually how low oxygen might be influencing the resilience and colonization patterns. So here I have community composition plots at only Kippo's on the in situ rocks, on the colonization rocks, and on the transplanted rocks. What we see with the colonization experiment is that within 3 months, these snails and limpets were able to colonize these rocks, even though, again, these animals are not so abundant at Kippo's compared to Mountain 12. However, within 17 months, they completely disappeared. They were not on those rocks anymore. With the transplant experiment, from transition to active, it is really hard to explain what's going on. We were super excited for this experiment specifically because this is the idea of a seepage expansion. So we're moving from a transition to an active site that is an increase in seepage activity under low oxygen conditions. So for us, this would be mimicking a little bit what would happen in the future with both ocean deoxygenation and ocean and methane seep expansion. But like I said, we don't really know what's going on here. These sea cucumbers, they're small sea cucumbers, were super abundant on this rock, and we don't know where they come from because we didn't find them anywhere else on any other rock, but they're there, probably taking advantage of the substrate and the food when no other animal would. For the transplants from active to transition, we saw that this community actually looks very similar to the transition community, showing us that the community at transition site Equipos was able to change and adapt to that new seepage activity level. However, we saw that densities were always lower after transplant, so it didn't actually recover that well. And there was an even higher reliance on photosynthetic production after transplant. And there are a few possible reasons to explain all of these observations. First, we know that low oxygen limits larval recruitment and connectivity. So this could explain why animals are not colonizing these rocks and showing up on these experimental rocks. It could also have been a case of predation of juveniles. So the tuna crabs that are super abundant at Quipos that I mentioned with the video could have been eating these snails and limpets, and that's why we didn't find them within 17 months. It could also be a lack of microbial biofilm and microbial establishment. So this is interesting because it would be the case that low oxygen is not only influencing the animals, but also the microbial community at Quipos. And because the microbial community is what is fueling this entire ecosystem, this is very important for the capacity for these ecosystems to survive a low oxygen conditions and possibly ocean deoxygenation in the future. So now you know how these animals how dynamic these animals are with all of these environmental variables and how they might respond to changes in those environmental variables. And you might be thinking that methane seeps are this very localized ecosystem, very specific ecosystem in the deep ocean. And, you know, why should we care about these communities and these ecosystems and these small animals that I've been showing you? And I hope that I can explain to you now that these ecosystems are not only like small islands in the deep ocean. They're not completely disconnected. And there's actually lots of links between methane seeps and other chemosynthetic ecosystems. To do that, I'm going to bring here two study cases that I was involved in. Um, yeah. So just a recap of what chemosynthetic ecosystems are, because now I'm going to introduce other chemosynthetic ecosystems. So they are regions in the deep ocean where we have chemosynthetic production, where we have microbes taking up chemical compounds from the water or from the seafloor to produce organic matter, to produce this chemosynthetic production that fuels these entire ecosystems. Chemosynthetic ecosystems include methane seeps, hydrothermal vents that you might have heard of them, whale falls, and wood falls. Whale falls and wood falls are also called organic falls. Some scientists also consider oxygen minimum zones as chemosynthetic ecosystems because there's been some report of chemosynthesis happening at OMZs. But I want to focus on these four here. So methane seeps, hydrothermal vents, and whale falls and wood falls are found worldwide, as you can see here in this map. This is actually not even updated. This is from 2011. But methane seeps, like I mentioned, are found on continental margins. So you're seeing here the blue little areas. Hydrothermal vents are found on mid-ocean ridges, so you see here in red, and they're associated with tectonic activity, while whale falls and wood falls can happen anywhere in the world where there's a river mouth that can bring wood from terrestrial ecosystems into the ocean, or where there are whales. Wherever there's a whale, the whale can die and it can sink to the deep ocean, forming these whale fall ecosystems. And a lot of the scientists started studying how these different ecosystems are connected. So there's a lot of animals that are found only at methane seeps, only at hydrothermal vents, only at organic falls. So those would be the organic fall specialists. And scientists started looking at how does this happening across ocean basins? How are these ecosystems connected across this? And then scientists noticed that there's also um, biogeochemical variables or the biogeochemistry of these ecosystems are also very similar, not within just one ecosystem, but among all of them. So, how is it possible that a species that lives here at this hydrothermal vent is also found here on a different ocean at a methane seep? And then, most of all, how are they also on whale falls and wood falls, which are completely different? So scientists came up with this idea of stepping stone hypothesis. So the stepping stone hypothesis basically says that whale falls and wood falls, which are just ecosystems that are not fixed to specific locations like methane seeps and hydrothermal vents, they can actually happen anywhere where there's a whale and a river mouth. They serve as stepping stone for the larval dispersal. So these animals that we're finding across all of these ecosystems, they have a larval stage that goes through shallower waters and shallower currents. The currents can disperse these larvae across the ocean basins, and they can use and settle down in whale falls and wood falls to reproduce and then produce more larvae, and then the larvae can keep going on and on. So this would help these animals disperse. So if you think that an animal has to go from all the way up north to all the way down south, if there are little stepping stones, organic falls in the way, it is more possible that these animals are going to survive little by little and going all the way to the north side of the world. Mhm. There you go. And there are a few ways that scientists can study them. But one of the things that we do is colonization experiments. So we deploy rocks, whale bones, and wood pieces in the ocean in various parts of the ocean. And here I'm bringing a few examples of these experiments that can go from 10 months all the way to 7 years. What's interesting about these studies is that they all show that substrates are promoting biodiversity in the deep ocean, and that there's also an overlap in animals across all of these types of substrates. And I'm going to talk about two of these experiments that I was involved in. So one is we deploy bone and wood at Mount 12, so the methane seep in Costa Rica, and the other is when we found this little shrimp here associated with wood and whale bones. So first, in Costa Rica, when we were deploying the colonization rocks that I mentioned already, we also deployed wood and bone in the vicinity of Mount 12 at active, actually, and transition sites as well. And this is not something that is just crazy scientists doing crazy things. This is actually something that can happen naturally. And we actually found natural wood falls. So these photos here are natural wood pieces that we found close to Mount 12, Jaco, and Quipos. And this is the skeleton of a swordfish that we also found super close to Quipos landslide. So this is naturally happening off Costa Rica. And we then manipulated this by deploying the wood and the bone at Mount 12. And there's a couple of things that were super interesting about this experiment. So here I have the photos of those substrates that we deployed. The top ones were deployed at active sites, and the bottom ones were deployed at transition sites. These photos were when we recovered them. You can see here that just from looking at the photos, you can see that these communities look similar. We have the Yeti crabs, we have snails and limpets, we have the giant mussels, that is one baby mussel here on the bone. So we see that at active site, seepage activity seems to override the substrate influence at these locations. So these communities look more like a seep community rather than an organic fall community. At transition sites, so now we're looking at the bottom photos, you see that they look different. And the communities actually resembled natural organic falls than seep communities, although we still saw some seep animals using these substrates. The other interesting thing about this experiment was that we observed a lack of organic fall specialists on all of these substrates. So the organic fall specialists are Xylophaga. This is a bivalve that lives inside the wood and eats the wood. and Ozdex, or they're also called zombie worm or bone-eating worm. So these are specialists of organic falls because they're feeding, excuse me, on the whale bone. And we didn't find them. So we hypothesized that the seep fauna can use these organic substrates both at active but also at transition sites and possibly to disperse and use them as stepping stones. But that seepage activity might be toxic for these organic fall specialists. And this is super interesting if we consider that seepage actually might be expanding because of ocean warming. So, how is that going to influence the organic fall communities? Are we not going to see more xylophic and Ozdex anymore in the future? The second study case that I want to show you is when we found this little shrimp here off Brazil. So when I was still in college, we deployed these whale bones and wood pieces in these landers off Brazil at three different latitudes and two different sites of two different depths, sorry. And we found when we recovered these landers, we found this shrimp. So this shrimp is called Elvinocaris moricola, and this shrimp was known only from methane seeps and hydrothermal vents before. So this was actually the first record of the shrimp associated with organic substrates. It was also the first record of the shrimp on the Southwest Atlantic. So this shrimp is actually known for methane seeps and vents in the Gulf of Mexico, Mid-Atlantic Ridge, and the East Atlantic. So these 3 points here are what we call the Atlantic equatorial belt population. And when we saw that we got these animals, we were super curious to see where are they coming from? Are they coming from these methane seeps and these hydrothermal vents? So we did a genetic study to see if there was a connectivity among these organic substrates that we deployed and these seeps and vents in the Atlantic equatorial belt population. And what we found was a low genetic distance. So they were not so different from the Atlantic equatorial belt population, but we did see a recent population expansion. So our animals, they were not so different from the other animals genetically, but they were still a little bit different. We showed us that they didn't come directly from the Atlantic equatorial belt population. They came from somewhere else first. The other thing that we saw was that most of these, actually all of these specimens that we collected were adult, and a lot of them were gravid females. They're full of eggs. Considering the lifespan of these animals and the duration of our experiment, we think that these animals crawled away from a closer seep or vent ecosystem because it wouldn't be possible for these animals to have reached our substrates as larvae and get to mature and reproduce and have, little larvae of themselves. So our observations showed that there are possibly undiscovered seeps and vents off Brazil. Um, we still haven't found them yet. My collaborators back home actually are still looking for these methane seeps. They still haven't been found. But what's interesting about this research is that it really shows us that, these ecosystems are definitely connected. And there are even connections that we're not even aware of yet. And there are things that, ecosystems that remain to be discovered in the world. So let's conclude. First, we saw the invertebrate communities at methane seeps are very dynamic and they change across environmental gradients, so seepage activity, depth, and oxygen. We also saw that they respond to natural changes in seepage activity. So naturally, seepage activity might change either because of earthquakes or because of climate change, and the community might respond to those changes really fast, actually. However, these environmental conditions have a role in defining colonization and resilience ability. And oxygen and low oxygen is going to have an effect in the natural community and limits the colonization and resilience of these animals. We also saw that the fauna responds differently to changes in seepage activity, depending on the availability and reliance on chemosynthetic production, that again is associated with seepage activity and death. And finally, we saw that seep fauna can use organic substrates as attachment and for food access, so they can use these organic falls as stepping stones to disperse across ocean basins. But seepage activity might be toxic to these organic fall specialists. And so what? Why do we care about all of these observations and conclusions? Well, we're definitely seeing that the deep sea does not look like that first video that I showed you. It is very diverse, and these hard substrates are really supporting this diversity of these animals. They're providing attachment, food, refuge, all of the things that I mentioned in the beginning, and really increasing the biodiversity of the ocean. We also with the communities that are being dynamic and resilient, if you think about climate change, ocean deoxygenation, ocean warming, but also direct disturbances like fishing and oil and gas extraction, it might be that these communities could survive all of these changes because they're very resilient and very dynamic. However, it seems that they are affected by low oxygen. So, in the context of ocean deoxygenation, this brings a new perspective to the resilience of these animals. So, it might be the case that when the OMZs really do expand, these communities might not be as resilient as we thought to the other disturbances like seepage expansion and direct resource extraction. And finally, I hope that you saw that, these communities are not isolated. It's not that, I'm bringing up again these maps to show you that they are found worldwide. And even though methane seeps are only on continental margins, the other chemosynthetic ecosystems are, within ocean bases, in the middle of ocean bases, and happening all around the world, and they're all connected. There's this huge, large scale across ocean basins network of these animals. So I think that it's important to think also in this context that if anything happens, if there is one of these ecosystems that might be jeopardized. So, for example, if we think about deep sea mining, at hydrothermal vents, or deep sea fishing and methane seeps, if we disturb one of these dots, the entire network might be affected, and we might see actually an effect on the diversity across the entire ocean in these chemosynthetic ecosystems. So with that, I want to say thank you all for listening. I'm happy to take any questions. I know this is might have been quite heavy on the ecology and on animals, but I hope that now you understand why us scientists care about methane seeps, so that next week we can talk about why and how people care about methane seeps.
Oh, thank you. That was great. A lot of compliments already coming in, so that was excellent. Thank you very much. Making
Yeah,
as was stated in the chat, a very complicated issue to us anyway. Very well presented and understandable. So
thank
uh,
you.
we'll take some time for questions and comments. John, go ahead.
So, did you choose Costa Rica because it was not disrupted in the ways that you mentioned, like deep sea trawling? Did you want to find the most natural place for that?
That is a really good question. And unfortunately, I was not the one that chose
Oh,
the location. I came to the lab and the project was already going on. But I think that the professors that were involved in the early stages of this research. So this project actually started in 2009. So our lab and our collaborators have been going to Costa Rica for many years now.
oh,
And I think it was probably a matter of, we find these methane seeps across these environmental gradients. It's super interesting. And they kept finding every time they went there, they found new things and new questions started, coming up. So I think it was a matter of we have these unexplored method at the time that were completely unexplored methane seeps. They had to collaborate it in Costa Rica and it just happened. And then I think that at first the experiments were not in the plan. They were just, collecting the natural things, the natural in situ rocks. But then I think that with time, and with the questions that kept coming up, and I think also, with this better um, idea of climate change and ocean deoxygenation and methane seep expansion, they started thinking about the experiments as well.
May I ask the second part? Do you
Yeah.
know if anyone who has done a study where there has been a serious disruption,
Yes.
like
So
trawling?
yeah, so we were actually also involved in a project in New Zealand. New Zealand has so many methane seeps, and there's a huge bottom trawling industry. So in 2019, I went on a cruise in New Zealand with some of our collaborators, and we were sampling at locations that we saw active trawling marks on the bottom. So they also actually, the research is still going on. We're not involved in it anymore, but they have locations, methane seeps that are undisturbed, that are actually protected, I believe, and seeps where they are disturbed. And I think that the idea is also to do experiments to see if this fishing disturbance is also influencing the capacity for these animals to recover.
Thank you.
Mhm.
Any other questions or comments? I actually, I'll go because I have some. If I understood well, it looks to me like there's obviously going to be changes in the ecosystem and migratory aspects as well. So how does this have the chain effect with predators of these animals, and what is the likely impact there?
That's a good question. And I think that we usually don't really talk about predation and predators of these animals, because I think what happens at methane seeps is a lot of animals that are actually there for food. So the fish, actually, right? And a lot of crabs also use methane seeps for food, for feeding grounds. It could be that we might see a difference in those communities. So to give an example, um, I think that it's okay, let me rephrase this. If we think that seepage is expanding, right, that we might actually see more methane seeps in the future. For these animals, for the fish and the crabs that are going over there and eating, for them, that's great. They're going to have more food, right? Um, but the thing is that this is not only the case. It's not just seepage expansion. It's not that we're only going to see methane seeps everywhere. But with ocean deoxygenation, we might actually see the opposite. And the seep might actually be worse, because of low oxygen, limiting the larval recruitment, limiting the colonization of these animals and the ability of these animals to really create this entire and thrive in this ecosystem. In that case, then this would be bad for the predators, so for the fish and the crabs. Um, so I think it's hard to say exactly what's going to happen. I think there's like good things, pros and cons on each side. But it is very complicated, actually. And I think that, we I hate to say it, like scientists are always saying we need more research, but we do need more research to try to see exactly how this might play in the future. But it is a complicated science. Yeah, for sure.
No, thank you. Anyone else? Erwin had a comment in the chat if he would like to say something. There he is.
Well, not really. I just made a comment how there's through your presentation this whole wide world of diversity that is very impressive. I can't say that I followed every logical step of your work. And I frankly don't want to go more deeply into it. I'm happy to know that somebody else is. But I made a comparison with one of the most marvelous books that I've ever read. If anyone here hasn't, I highly encourage reading Entangled Life by Merlin Sheldrake, which is all about fungi. And to me, it was the subject of a local book club here, and the impression was shared by many that this is opened up a whole new world for people that they had barely aware of existed. And so it's a bit similar with the pictures that you showed us. What I'm less clear about, you allude to it at the end, is exactly what all that implies. How does it change anything that we are or should change anything that we are doing or not doing, apart from the obvious, like stop trolling the ocean floor. Well, to be honest, I don't need your analysis to say this is such a terrible idea. Let's not do that.
I appreciate you.
What the value added for human behavior is of your study is not completely clear to me.
Yes, that's a good point. And I think that we're going to be able to talk more about that next week. I will say that. So next week that I will be presenting about my economic valuation, I also discuss a little bit in the paper about how that changes our perspective into the deep sea and also how, we're going to bring all of that science to the people and what are actions that we can take as society to, make changes about it. And not only, in terms of conservation and things that we can do as society, but also in terms of why also we should be doing all of that, I guess. So, yeah, I would say that we'll talk more about it next week. And I appreciate you saying that, you don't need all the science to know that this is an important ecosystem and that we should protect it. Because I think that sometimes, some people don't see the deep sea as this important ecosystem in the ocean because it's far from reach. It's not something that we can see with our own eyes. So people think that it's just, if you don't see it, it doesn't exist. So I really appreciate what you said that, you know that it's important and you know that we should be, taking care of it.
Thank you very much.
There anybody else that would like to ask a question or a comment? Okay, I'm not seeing anyone. So I think next week we'll see probably more of what we're familiar with
Yes.
with the economic and human connections with the deep sea. And but this was extremely interesting and a lot to think about and the importance of the ocean. I think we all know, but now we understand the importance of the deep sea aspects where we normally probably think of the upper levels. So
Mhm.
we look forward to next week and thank you very much, Olivia.
Yeah. Thank
See
you
you
so
next
much.
week.
See you next week. Bye.
Bye bye.